So the recommended analyses, so here's kind of the list of recommended analyses from the adverse event white paper. However, the adverse event white paper doesn't really go into complex study designs or multi-period studies. So some of this is a little new from the white paper. You can't get all of this from the white paper. But the first column is directly from the white paper. What do you do for that treatment period? Study drug versus placebo. So we have an overview of adverse events. We find that handy to just have a quick summary of adverse events. Treatment emergent adverse events sorted by preferred term within system or in class. Treatment emergent adverse events sorted by preferred term without regard to system or in class. Common TEAEs, TEAEs by max severity, and AEs leading to study treatment discontinuation, and SAEs. So hopefully that package sounds familiar to you, and hopefully that's what most people do. However, we have seen variations to this, right? So we have people who do things slightly different, but you would think by now, as an industry, our goal could be, can we at least do that consistently? Can we at least do that list pretty similar and have the same look and feel of the display so that our core clinical reviewers don't have to review this very simple package in all sorts of different ways. Okay, so what is a little new is this, what do you do now for signal detection purpose? What do you do with the study design like we talked about? Now you have this extension, and now you have a follow-up period. So our recommendation is that you look at your drug, right, your study drug, and go and look, just count your events, right? Count your events that occurred while on study drug, and then have another column that looks at study drug that includes the follow-up period. Now this set doesn't have a control, so it is a little bit smaller list, but we do think overview makes sense, TEAEs makes sense, AEs leading to discontinuation, and SAEs. Okay, so let's talk a little bit about one of those details that can drive people crazy. So in a study like that, how do you define your time period, right? So what is the treatment period? Is it, do you do it by visit? Do you do it by dose dates? Do you end at dose? This is one of those things we vary on. The most basic analyses, adverse events for a treatment period, we have variations on how we handle the time period of interest, right? And it's a silly thing to vary on, but it is not an easy thing, and it's not certainly obvious. So the recommendation is that your treatment period does start at dose, right, when you start taking dose, and then go up to when the next dose of the open label extension. And part of these recommendations are based on trying to not end up with gaps, right? You don't want, if you do kind of stop at the end of the treatment period at that visit, you could have that time between then and getting open label dose, and you could have an event in there, and then is that somehow could get missed. So a lot of these are trying to avoid any kind of gap so that all your events are at least part of a reasonable summary. So another, let me see if I talk about it later. Okay, so for the study drug period, the idea is it would count all events that occur while patients on study drug. And then the next column would be study drug, and then including follow-up. So this would then, again, the details can vary, and this is some of these variations we're trying to address, is that you would kind of start your period at the time of dose. So that way, if you count an event during study drug, you're not counting one that maybe it happened before, slightly before. You don't really want that in your summary. Okay, so here's the shell. This is in the white paper. You probably can't see it very well on the screen. So this is in the white paper, and this is one that it should look very familiar to you. You have, we just assume you have a low dose and a high dose and a placebo. And you'll see these other columns, though. We do recommend more than just giving little n in percents, right? We think it is worthwhile to put more on your table to help with the signal detection process and looking for magnitude of effect or how strong your imbalance is, right? So we do have a big discussion on confidence intervals versus p-values. a risk difference or odds ratio, but the idea is at least have a measure of magnitude effect. This mock has risk difference, and then this mock ended up putting confidence intervals, but certainly we'll talk a little bit more about p-values as a possibility. But this has an idea of how strong your balance is, and it has a measure of magnitude of effect. So if you're one of those people who just have little n's and percents, we would argue that we would promote that you do more than that to help facilitate the purpose of the table. Okay, then here's one with maximum severity. This one just has some summaries. We do look at mild, moderate, severe. Now, it's very common in oncology to look at CTC grades. That's fine. You would just have the grades instead of mild, moderate, severe. We do have a paragraph in the white paper about pros and cons of just having a subjective mild, moderate, severe versus CT grade-based severity levels. We get that question a lot. It's like, well, why doesn't everybody have CTC-based severity levels? Because they're objective, they're not as subjective. And our position is that we don't necessarily think that's necessary. We think it does help in oncology, but we don't think it's necessary. But this could evolve. I mean, it'd be interesting to get other opinions as this evolves. But the idea is we don't think we need it because we already have AEs leading to discontinuation, which is an approximation for how severe something is. We also have seriousness, right? We have many ways to look at severity, and do we really need to add burden to all of our trials to have an objective way to look at, mild, moderate, severe, and so forth. So we have a discussion on that, so we do include some of those things. So this figure is something we do recommend. It is in the white paper, and we are trying to get people to move from tables to figures as much as possible. We don't take the position that everything has to be a figure. In most of our recommendations, we still have a lot of tables. So we're not giving up tables, and nor are we trying to say everything needs to be a figure. But we do know there's a lot of interest in having more figures, and people are frustrated with looking at long tables, pages after pages. So we are trying to evolve the field to have more figures. This one is a figure that was another group that's involved in safety graphics, put together a lot of figures. We got this from CTSpedia, so there's something called CTSpedia. If you go there, there's a lot of safety graphics, and this one is there too, so it's not like we made this one up. I mean, this is one that people have suggested for a while, but we want to help promote it to become predominant practice, right? So you have all these people, suggesting certain graphics. I think the role that we're trying to bring is to say which of these graphics should we make predominant practice, right? So this is when we say it could probably start becoming more predominant practice. And it's for common TAEs. It has percentages on one side and confidence intervals on the other. It really does help rank which events are most important for your medical colleagues to look at. Okay, and for the table for the all-study drugs, so this is the table that could cover the entire study for any event that occurred while on study drug. You don't have a control. You have the one column that says here are the events that occurred on study drug, and here are the ones that occurred on study drug or after. So it can kind of give you both look. The latter column is just a more conservative way of looking. We're just going to assume anything that happened after drug attributed to drug. But most people don't want to just do that. They really want the summary that looks at it. Hey, what is it if they were just on drug? But then they also kind of want, well, now if I assume that everything after is drug, what does that look like? So that's why we do recommend a 2-column table. Okay, so let's get into the rationale a little bit. So the study drug versus placebo, this is when we have a control that's all related to that signal detection, that ADR determination, the kind of things that are important to know for determining whether an event is an ADR. So those tables provide you that strength of evidence for an imbalance, and either you have your percentages, you have your risk difference or odds ratio. You may choose to have you have a confidence interval or a p-value, and then it also gives you the magnitude of effect. So those are 2 key parts of ADR determination, and that's the purpose of recommending those in those tables. So the other tables that look at the uncontrolled data, this is a chance for medical to look to see if there's anything rare or unusual that they wouldn't expect in the population, or sometimes there are events that are probably related by the drug, just by nature of the event. If they see it, it's probably due to the drug. So they look for those kind of things, and it provides an easy way, right? So it doesn't, this gives a way to look at those things that are more rare without having to go through a whole bunch of tables, right? So if you made a table of study treatment, an open label table, and a follow-up table, you have 3 tables with all your events spread out, that's not very easy for the purpose of those tables, right? So it's really key to understand what the purpose of the table and try to figure out how best to display that. And that is one of the things we sometimes see, is a lot of these events are chopped up into different tables, and that's not always very helpful. All right, so the overview is, sorry if you can't read it, I'll read it for you, but the overview is recommended because it does provide a nice summary for a study report of the kind of events we see. It usually contains TEAE, serious, number of patients with a serious event, number of people who discontinued. It's a nice summary of your adverse events. Sometimes we get asked this about way to sort, right? So unfortunately, we're still in a static world, right? We talk about static displays. As much as we would love to get to interactive displays, and I think we're moving in that direction, which is good, and we're all supportive, our white papers are still, they're providing recommendations of what we do today, what we would recommend today, which may not be what we would recommend a few years from now. But today, while we're still living in this static, digitized paper world, we do recommend treatment emergent adverse events sorted in 2 different ways. Most of our colleagues find this helpful, but sometimes they do want it sorted by preferred term within system organ class, and then again without system organ class, because without system organ class you can really see the most common events pretty easy. So that's the only place that we say we think we need to sort things both ways. We don't think serious needs to be sorted multiple ways, or AEs leading to discontinuation probably doesn't need to be sorted multiple ways, but these are big tables, and while we're in the static world, but people can't interact with the data, we do find both of those helpful. So the common TAEs is something that hopefully someday we no longer have to recommend. So as you saw that ADR determination, was common even on there? How common an event was? It wasn't. It's more about your imbalance with placebo, your magnitude and effect. Common, you're going to get your headaches and your nauseas, your colds, your flus, whatever, right? So common doesn't really have a huge role in signal detection, yet it is our habit to always show common things, talk about common events, and that's something I think we would like to change, but we would not recommend that you do away with this table or figure today. We're not there yet, right? So our recommendation in today's environment is that you probably do need to create a display of common TAEs. Of course, it's that figure now, not the table that we like. So even though it doesn't have a big role, it's in everybody's templates. It's in all the. It's kind of one of those things that we just have to produce until we get to influence the templates, until we get a chance to change how CSRs are written. Treatment emergent events by max severity. This is something that is also used for signal detection in addition to characterization. And this is something not everybody understands, right? You might have to help or remind your writers, remind your medical colleagues that it does have a role for signal detection. So it is there because you might miss a signal with all your severities kind of mixed together, right? So the idea is if you just look at severe, it's possible you will see a signal that you would not have otherwise seen by having all the severities together. So most people, when they write about that table, will say, well, most events were mild or moderate, right? But they forget that this actually has a pretty good big role in signal detection as well. And we should be saying, well, when we looked at just severe events, we didn't see any additional events of interest or that required further screening or something like that. We forget that purpose of that table. AEs leading to study treatment discontinuation, same idea. This is kind of a approximation for how severe it was. If they actually had to quit drug due to that event, that's kind of a bigger deal than just having the event. And that certainly does help with signal detection. And serious events also helps with signal detection by focusing on the more severe events. All right, so I already alluded to this, so this is a common pitfall, right? So it is a common pitfall to forget that this maximum severity table has this role in signal detection. And there is a really nice discussion of this in the 2010 FDA clinical review template. That was one of the things on the pre-read side that I thought was quite useful. They actually have a really nice discussion on how you can miss signals by focusing on, if you just put all the severities together, you might miss something. So I would encourage you to read that section too. All right, so let's move into some frequently asked questions. So one of the questions we get all the time, and me too, is how is treatment emergent defined, right? So this is another one of those areas that it seems silly that here in 2019, after our industry has done hundreds and hundreds of clinical studies, we have variation. We have a lot of variation in how people define treatment emergent, right? And we do have in our analysis and display white papers project team, it is our goal to have a white paper just on this topic, right? It's such a big source of variation. We thought we could fit it in our adverse event white paper, but there was too much to that, so it's going to be its own white paper. So the adverse event white paper, even though we didn't go into a whole lot of detail, there is a section that we do have a brief discussion on where we see some of the variations and some references that go into this a little bit more. But our hope as a project team is to get to consistency on how we define treatment emergent. So that isn't our goals, but hopefully over the next year or two we can try to get to some recommendations on that. So it is out of scope for our conversation, but one of the common pitfalls is assuming that your way of defining treatment emergent must be the way everybody does it, right? I think your average person doesn't even know the extent of variation that we have on this, right? I think people go, well, don't everybody define it this way? It's like, well, no. So and not only is there variation in definition, there's variation in collection, which makes this an even more difficult conversation to have because the collection is very different from company to company.
Even within a company?
Within a company, I would say most companies, there's probably not a lot of variation, but there could be some. I would say it's mostly between companies where we see the variation. Yeah. Okay, another frequently asked question is why do we count number of patients with events instead of number of events? Well, there could be some adverse events of special interest where absolutely counting events makes sense. But for signal detection purposes, for the methods that we applied for our entire treatment emergent adverse events, it works fine. Counting number of patients seems to work fine in most cases. We might have some exceptions to that, and there could be some adverse events of special interest where you wouldn't want to do that. And that really happens to events that basically are going to occur in every patient eventually. Right, so some people say maybe hypoglycemia and diabetes. Well, maybe if your study's long enough, almost everybody might have some type of event where it's more interesting to count how many events. Infections might fall in that too. But those will become adverse events of special interest. But for your run-of-the-mill event, where you're just looking for events that require further scrutiny, percentages seem to work just fine. The other thing that causes some problems with this is the collection. We talked about collection can vary. Well, somebody at the site might record every single headache someone has, right? And someone else might say intermittent headaches, right? So they might write down, though, they have headaches all the time. Well, that, from a statistical point of view, how do you count that, right? Intermittent headaches is still one if they record it that way. But the people who are recording each headache, well, then that one we can start make some sense out of it. But that collection variation does hamper our ability to count events instead of patients. All right, so why is that recommended list for during study drug and during study drug after shorter? All right, so why did we recommend fewer things? Well, the control data we recommended common TEAEs. Well, why would we not recommend that for study drug? Well, remember the purpose of that is to help medical look for the more rare things, right? The things that are going to catch their attention if they think, oh, we shouldn't really be seeing this many events in this population, right? So there's no reason to have common for that set, right? So the placebo-controlled data has the best is to look at for common events. You should look just there. Having common events for your all treated group doesn't have a purpose, right? It's not serving a purpose. And max severity, at least with signal detection, was also looking to see if you have an imbalance, would that be different if you just focus on the severe event? So that's also relevant just for control data. Now, with the uncontrolled data, you might be interested in that for characterization purposes, right? If you have a rare event that ends up being considered caused by the drug, well, you might want to talk about whether they were severe or mild or moderate, right? But that then gets to characterization, and you would do that if you need to. But from a signal detection, there's no purpose for that, the one that has all the events. And time to study treatment discontinuation due to AE, that one is also not as useful without a comparator. So that's why we do recommend different things on whether you have a control or whether you don't have a control. All right, so why isn't dose in that table, right? This is a very common question or common request, right? They want to take that, so the study design, if you remember, the treatment period, low dose, high dose, placebo, and then the open label extension had just high dose, okay? So why don't we just have one big table that had low dose, high dose across the whole study? Very common pitfall. People think that creating summaries of dose across your treatment periods actually is giving you something meaningful, right? People think that helps. But the problem we have is that it's an extremely biased comparison, right? So there's only certain people who are making it to that extension phase. So it might help thinking through a titration, just to get the point across. Let's think of a slightly different study design, where it's a titrated study. So everybody's on a low dose, and after a couple of weeks go a little higher, after a couple of weeks go a little higher, right? That type of design. If you summarize by dose, guess what? The high dose looks amazing. The high dose beats low dose and middle dose, and you're gonna come out thinking that low doses and middle doses are safer than the high dose. Well, that's certainly not true. It's an artifact of the design, right? So there's a bias, right? It's only people who can tolerate the drug who are even getting the high dose, right? So that's a very significant bias. And that's why we really need statisticians and medical to work together on a lot of this cross-functional analytical planning, because all of this kind of stuff matters. And some people, statisticians, we tend to be a little bit more in tune to looking for what could be biased, and we need to educate on this. And this is one of those things that seems very common. We get asked for this even in integrated analyses too. They just want a simple study by dose. Well, guess what? Your simple summary by dose. Well, your simple may not be good enough, right? It could be extremely biased, and we've seen real examples of this where it really is not helpful to have, to look at dose for these designs where dose changes all the time. Okay, so why some companies do just give adverse event tables, little n and percent, right? That's all you get, little n and percent. Well, big N too. But why would we recommend confidence intervals or p-values? So certainly, as we talked about, having a measure that assesses the strength of evidence for an imbalance is helpful, right? That's a helpful thing to have. And one of the biggest criticisms for not having p-values anyway is to say, well, for safety, we were empowered for safety, we shouldn't be doing hypothesis testing for safety. That's all true. However, that's for most people who do use p-values in safety know that. They know that it's not hypothesis testing. It's still a number between 0 and 1 that gives you an idea of how strong your evidence is, right? The closer to 0, the stronger it is. The closer you are to 1, the less strong it is. It's a number between 0 and 1 that tells you how strong your evidence is, and some people find that very helpful. Confidence intervals can do that, give you an idea of how strong your evidence is as well. Those are what we chose for the mock tables in the paper, but just so people understand that for those people who do like p-values, they find it very convenient, right? It's even more convenient sometimes in a conference middle to kind of rank in order of which events have the highest evidence for an imbalance, and a lot of people find that very helpful. And we're going to continue these discussions in our white paper group. There's really cool figures. There's a really cool figure called a volcano plot that uses uses p-values on one side, odds ratios on another, and those are really cool plots. And but we do need to educate that when you do have a p-value used in some way, it is not hypothesis testing, it is just simply a measure of a balance. And so it is an educational thing that they are okay to use, just don't dichotomize it. Don't say if you're less than 0.05 it means something, and greater than 0.05 it means something. It's used as a continuous measurement for safety. Okay, let me cover real quick. A common pitfall is if you do use p-values or confidence intervals, it is common to try to use them to say whether or not something is significant or not, and that practice needs to stop. And I think statisticians need to help that practice to stop. We can't, whether it's a confidence interval or a p-value, that we need to get away from referring to something being significant or not, or a confidence interval including 0 or 1. All of that doesn't have a big role in safety. So that's something we all need to work on. Also, that a lot of people don't necessarily understand, like your medical colleagues, may not understand, what is the difference between imbalance and magnitude effect, right? Some people might think they're one and the same, right? So this is an example that you can share with your colleagues if they're confused on what the difference is on strength of evidence and magnitude. So here's different data. The odds ratios are the same, so the magnitude of effect is the same, but your strength of evidence they are not the same. So that's why we encourage both. We think both of those measures have value in trying to help decide whether an event is caused by the drug. Right, here's this volcano plot I spoke of. So this plot is currently not in the adverse event white paper as a recommended display. We briefly mention it. It's something we might consider for the future. But the main reason why it's not recommended today is because it's not a very great figure to fit on a page and have it be static, right? So this figure, so every dot is a measure of preferred term, so it's an event, and it's plotted against its p-value, or number between 0 and 1 for strength of evidence, and then odds ratio. So the events that kind of fall in the top right quadrant are those that are probably the ones that deserve the most scrutiny or attention. So this particular plot, they were able to go ahead and put the preferred name on these, but in real life, with a lot of events, you may not be able to do that. So in the static world, this is a very difficult figure to really interpret, right? You really can't name every single dot. In the interactive world, right, when we can actually have some point and click and hovering, and you can hover over a dot, see a preferred term, all of a sudden we're going to love this graph, right? So we're anxiously waiting, to get some interactive figures to actually produce these in real life in a study report and a submission. And we do have a project team within Fuse who's helping do that, right? So there's a data visualization project team, and we're testing this. We're testing one of these right now, or at least this year. We think we'll test one of these hopefully this year with a real submission, and we'll see how it goes, right? So there's a lot of work to be able to produce interactive displays, and once we can get that to happen, our recommendations might change quite a bit, right? Because right now the white papers are written assuming we're still need some static tables and figures, but we'll be moving, and that's why our white papers we consider, we're going to have future versions. They're not kind of done. We don't consider it done, right? Things will evolve. Our demographics, disposition, medications white paper has gone through a version change. We have a version 2 of that because there were some changes on what the FDA wanted for medication data. So that's already undergone an update. So adverse events may too, and we may end up recommending this figure. Okay, so why does post-baseline end at the study, treatment, disposition visit for those who discontinue? So I actually forgot to mention something about this design we're thinking of. Okay, so this design that we thought about, you have a treatment period, open label period, follow-up period. Let's assume, which this is becoming more and more common, that when somebody decides to discontinue study treatment, that they're encouraged to stay in the study as if they didn't discontinue. Follow the normal scheduled events, right? Don't get moved to a follow-up phase. You follow the normal scheduled events. So for this design, there's quite a bit of debate on how do you end treatment for those patients, right? Do just use the time as if they were on drug all the way through the treatment period, or should we stop when they decide to stop study treatment? Now, this is one of those difficult things that could vary depending on the study itself, and this is why statistical thinking and medical thinking have to come together and think about some of these things, because it does matter. Our general recommendation is you do stop at treatment, because when they stop treatment, because once they decide to stop treatment, they are usually allowed to take all sorts of other things, right? They can take whatever. And so let's just say the worst case scenario that your placebo patient stopped, and then they're taking these other drugs, they have all these nasty side effects, right? So now all those nasty side effects are going to get attributed to placebo, and guess what? You made it harder for you to find your signal, right? You made it more difficult to look for an imbalance between drug and placebo, because now you've created a whole lot of bias by all these events that these placebo patients are taking for other drugs, right? And we don't want to miss our signals, right? So that now, if it's a study design where those patients are required to follow the protocol and what they're allowed to take, they're not allowed to take anything, well, then we'd probably go the other way. We probably would go ahead and count those events. So conceptually, we want to be able to count the entire treatment period, but however, it's a balance of biases, right? You just can't, you can't just take that as a rule. You do need to think about what happens when they actually stop drug. All right, this is very frequent. Why didn't you add 30 days or some other time based on the half-life of the drug, right? There are people who have a rule of thumb that you must always add days, right? Add 30 days or add 2 half-lives after the treatment period. So what common pitfall is assuming that your convention or your thinking and how that should be done is predominant practice, right? So there's a lot of people who say, well, don't everybody add 30 days? Like, well, no. A lot of people think the way they're used to it is what everybody does, right? So, and that's not true. This actually has a whole lot of variation around it too. Now, this is another thing that depends on the study design, right? So in the design that we were looking at, would this make sense at all? No, right? You're going to take the 30 days of, so the placebo patients go to open label drug, and you're going to take their first 30 days of drug and attribute that to placebo, right? No way, right? So you can't just take these blanket rules and apply it. Now, somebody would say, well, we know that. We're not going to count those placebo, but we need to count it for drug, right? Well, we'll count 30 days after drug, but not placebo. Well, guess what? You just did something to one group that you didn't do to another group, which is another source of bias. So we really do not encourage this unless it's a very simple design where you might have, treatment and control that goes immediately to a follow-up. If you just do both periods on that, maybe that's okay. But you can't just take a blanket rule of thumb and apply it to every design. All right, so one of the common pitfalls is that for safety analyses, choosing the most conservative approach across various choices, right? I don't know how many times we've heard, well, we have to do it that way because we have to be conservative, right? It's safety, we have to be conservative. Well, guess what? Being conservative from a total count perspective, right, which most people think of when they think conservative, may actually make it less conservative when you're looking at comparing with placebo, right? So a conservative, the thing that gives you the most counts of something, right, doesn't always mean it's conservative when you look for your treatment versus placebo difference, right? And our purpose is to look for treatment and placebo differences. That's the main purpose. We're trying to decide what are adverse reactions of the drug. And so everybody's heart is in the right place, but it might not be the best choice, right? You have to think of the purpose of the table. And if the purpose of the table is look for imbalances with placebo, you're trying to make that as unbiased as possible, and it may very well mean that the most conservative choice may not make that be the most conservative, right? So you got to think of the comparison, not the total count. And we're trying to make that comparison free of bias, and that's where statisticians can really help. Okay, why don't you have analyses for the open label period by itself and the follow-up period by itself? This is a common pitfall, maybe not. I I don't know how common. I've seen it, certainly seen this tendency, because it's really easy in a statistical analysis plan to say these safety analyses will be done for each study period by period, right? So one single sentence, right? Very easy sentence to write. Well, that turns into 1,000 tables, right? Most of which don't have much value. But so there's this tendency to just want to simply say, well, I'm just going to do all these analyses by period. But that's not consistent with the purpose of these analyses, and you might end up with tables and figures that are just volume that doesn't really help get to the purpose. Some people think they want the follow-up period by itself to look at withdrawal effects. There are better analyses to do for withdrawal effects. You would want to definitely base some data, especially in the design. You got to think about these things, right? Especially in the design where people are encouraged to follow the normal schedule events, even though they're off drug. Some of those people could have been off drug for a long time in the treatment period. So if you use your follow-up period and try to do an analysis and think that's giving you something on withdrawal effects, you're sadly mistaken. You've got a lot of people who've been off drug, and if there's something with withdrawal effect, all those are going to happen in the treatment period, right? They're not going to wait until the follow-up period. So you need to, these are the kind of things you need to think about when you're planning your analyses. Why didn't you include exposure-adjusted incidence rates? So that's a common question. For control periods, we don't usually have differential observation time. You start thinking about exposure-adjusted incidence rates when you have differential observation time between your drug and placebo. That's not usually the case, so these are not usually warranted. And for the during study drug period, they might be useful. Their usefulness is still a little bit questionable because you don't really have anything to compare with. You might have literature to compare with, but then we might say, well, say that might be important for an average spend of special interest, but having a whole series of exposure-adjusted incidence rates for 1,000 events, I'm not sure that's useful. Not usually considered that useful. And we do have a lot of common pitfalls with exposure adjustment. Everybody thinks it's God's gift to analysis. It fixes all problems related to time, right? Now, think about that titration example I gave you, right? Low dose for a little bit, high dose for a little bit, or middle dose and high. You can, let's say they get on that high dose and they're on it for a long time, right? They think, well, if you just adjust for exposure, now you can compare those doses. Well, did that do anything to get rid of the bias that it's only people who can tolerate the drug that even get to the high dose, right? So adjusting for time didn't touch that bias whatsoever. So it is not the cure-all, for all analyses. And I think there's a lot of people who think it is, right? Think, well, as long as you adjust for exposure, everything's okay. Well, no, you still might have some biases you need to worry about. Another thing that was covered in the AE white paper, this was certainly some feedback. We had some from our FDA clinical reviewers, some feedback from them. This was one of the things they wanted to make sure we covered, is that it's really not appropriate to do an exposure-adjusted analysis that includes some off-treatment time, right? So let's say you want to be conservative and account all those events that occurred during that off-treatment time. Well, now if you take an exposure adjustment of that, you now underestimated your EAIR, your exposure adjustment incidence rate, is now underestimated because you included off-drug time, right? So the combination of off-drug time and exposure adjusted is not a good combination, and that should be avoided. Okay. Why do some compound teams use subjective severity levels, mild, moderate, severe, when CTC objective levels can be used? I actually kind of already snuck this in, so I won't say much more. We do have a discussion on this, and we do think for most indications, subjective severity is sufficient, given the fact that we have other ways of looking at severity. All right, so why didn't you include a summary of adverse events considered related by the investigator? This is something that I imagine some companies are creating, right? Some people probably have it in their habit to include a summary of events considered related by the investigator. This is one of those that we hopefully as an industry together kind of decide to no longer do, right? And I think it's easier to do it together, or we have a white paper that says we don't think you need it. It might be easier to discontinue something that you're used to doing. So a lot of companies are used to doing this. We think it's okay to stop. And we're in this together to try to right size our volume, because we sometimes assignments have a large volume of tables and figures. The clinical review template, FDA's 2010 clinical review template, it has a really nice discussion on this and why they don't consider it all that valuable. Basically, the assessments by the investigators are considered highly dependent on the information available at the time, like what's in the investigational brochure. So they think that's kind of biased and influenced by that. So they don't find it very useful at all. In fact, there's another cross-industry group, the science group, who basically advocated for stop collecting it, right? We all collect, or most of us, I think, collect it, right? They advocate to not even collect it except for series events, and there's a role for it in series events for individual case reviews. But for non-series events, there's that whole group says maybe we shouldn't even be collecting it. But we make it a habit to collect it, we make it a habit to summarize it. I think one of the things that's absolutely in our control is to at least stop summarizing it. We probably still need to collect it for the time being. We do have Japan labels, right? The Japanese labels tend to use that data for their labels. And right now we also have, there's EU layperson summary guidance that uses some of that data. So we have a couple little areas where this data is being used, but maybe someday we can get to the point where we're no longer collecting it. But we have a couple pockets to address before we would recommend not collecting it, but we're absolutely ready to recommend not summarizing it. Okay, so interpretation, we covered some of this already, but when you do have confidence intervals and p-values, we do know we don't want to say whether something is statistically significant or not, right? It is simply these are continuous measurements that give you an idea of where you are in the scale of imbalance and magnitude. If you aren't already aware, this is absolutely a hot topic right now with the American Statistical Association. They had a special issue in March of this year, and they just had an associated webinar just a few weeks ago, I think. Right, this is a hot issue that they're making a plea for people to stop dichotomizing p-values, right? So p less than 0.5. So if you're 0.49, you're significant. If you're 0.51, you're not. They want all of that to stop. They've been trying to advocate for this for a while, but it's getting kind of more serious and more vocal about it, and that's even for efficacy, right? Even for efficacy, they're trying to get away from this statistical significance language, right? So they might have a bit more of a journey than safety has, right? It might take a while for people to get used to not calling something statistically significant for efficacy, but for safety, we can do that now. I mean, I think there's no reason why we would stop that now, and some people already have. I think what's going on now, some don't do that, but then they stay away from p-values altogether or confidence intervals altogether. But that's not what the message is, right? They're still useful. Confidence intervals are useful, p-values are useful, but we can't overinterpret them. We can't dichotomize it. We can't just say, well, you decide one thing if it's 0.49 and something else if it's 0.51. A lot of good stuff, a lot of literature in that special edition, and it's kind of going to be a nice evolution, right? I had pulled this quote from one of the announcements. As statistical significance is used less, maybe we do have statistical thinking used more, right? So there's just more to the thought process than the dichotomizing of p-values. All right, so with the interpretation of the during study drug and during study drug after, so you just have a lot more time on drug, right? That open label extension period just gave you a lot more time on drug. It's uncontrolled data, so it's a little bit limiting on what we can interpret from that, but it certainly gives more of a chance to see events that could be a little bit more rare. It gives it more exposure so you can see it. So that interpretation is primarily driven by medical judgment, right? Knowledge of the population, and just knowing whether or not that event is a surprise or the number of events is a surprise. So very heavy on medical judgment. Okay. Another common pitfall is to identify a primary safety analysis set, right? So in the study that we just saw, somebody said, well, the treatment period is our primary analysis set, right? We say that. We have a tendency to want to say what's primary. We don't really need to do that, right? So unlike efficacy, we don't really need to define a primary. And And the common events, your placebo-controlled data is going to be way more useful than your uncontrolled data. But for your more rare events, guess what? Your uncontrolled data is going to be more useful than your placebo-controlled data, right? So you don't need to identify a primary. So if you see that in writing, you can, maybe say something. We don't need to do that, and it's kind of misleading. All right, so then the other common pitfall is focusing a discussion on common events, right? This is very common to focus discussion on common events, right? So I think we need to eventually get away from that and focus more on the things that show an imbalance and have high magnitude effect, which is where once we start maybe using that volcano plot, having those events that fall in that right quadrant is what should focus the writing, right? And to get away from always just saying, well, these are the most common events from the study. Well, I don't know, at some point you say, well, who cares? I don't know, right? The other stuff is more important. Right, so this is something that was highlighted in the update clinical review template, and it's very important for interpretation, is to be careful splitting, right? So sometimes sometimes very similar terms end up getting mapped to different preferred terms. That makes interpretation very difficult, because if the same things are kind of split using different terms, it's going to be harder to find your imbalances and your magnitude effect. And that is why we do recommend what we call clustering or consolidation. We don't necessarily think it's a little bit of a time-consuming exercise for a medical person to say, well, I really think these are the same thing, they should be analyzed together. We don't necessarily recommend that for every single study report, but we do recommend that when you do an integrated summary. Right, we need that. It's as brutal as that exercise is to manually look through, what events could be basically the same thing. We do need to look for those and maybe have another table or another analysis that has those events together, so we don't miss a signal. Same with lumping. Sometimes you can put too many things together, and they're very different things. So this, a lot of times, this really pertains to when, as a study is ongoing, and let's say the site just describes an event, and what happens is sometimes they don't describe it in enough detail, or it's too vague, and it might get mapped to some vague term or mapped to some term that doesn't really make sense. So this is really, it's a common pitfall to really, not really appreciate the importance that the medical group reviews this, right? To make sure that the descriptions that sites are saying aren't too vague. You don't want the queries to be misleading, say, don't you think it should be this event? But a query could say, wow, that's a pretty vague event, can you be more descriptive, right? And then hopefully it gets mapped to a more meaningful term. Both of these points are made in the FDA clinical review template and very important for reviewing adverse event data.